All right, hi guys. I'll be explaining LO1 group problems today. Um, so here we go. Alrighty. So we can see this one, what proportion of the offspring expected to have AA, BB, CC, DD, EE genotype. So we can go down here, we can match them. We just do individual Punnett squares. We just do individual Punnett squares. So you go to A little a after they've been calculated. So this is 1/2. You go to big B, big B, big B, big B is 1/4 because of the Punnett square and meiosis, which equals this. Go to DD, big D, DD. You can see that it's 1/2 through the Punnett square. You go to little EE. So little EE is obviously 1/2, which is going to give you 1/28th fraction. And then it's the same song with all these. You just do individual Punnett squares for the A's, the B's, the C's, the D's, and the E's. So again, you can go to little AA, which is 1/4 times little BB. Little BB is 1/4. You go to little CC. Little CC is also 1/4. Then you go to big DD. Big DD is 1/2. You go to little EE, which is 1/2. You multiply them together, you're going to get a fraction of 1/256. Then you do the same song with these. This one actually has a small dd, and this one there is no small dd with homozygous D's and heterozygous D little d. So this one actually isn't logical because of that. Then you go to number D, and you do the same song. You're going to get 1/64 fraction. So now I'll screenshot and pop up the other questions, and we'll continue. All right, here we go. All right, we're going to share this screen share. And now we're online. So here's number 2. You can see it's just fill in the blanks. If we go to A, paired homologous chromosomes are sometimes called. So you can call them homologs and also homologous pairs, which is just two names referring to the mom, the parental and the maternal chromosomes, paternal and maternal chromosomes, which are inherited from mom and dad. And they're pretty close to identical, but not quite. The alleles are different. And they're the non-replicated, non-sister chromosomes. So these are just the two other names for them. Number B, the structure that connects a centromere to the microtubule of the meiotic spindle is called kinetochores. So once the centromere has connected the homologous chromosomes and also sister chromosomes into connecting chromosomes, then the meiotic spindle can connect to the kinetochore. The kinetochore is just as a whole the centromere is connected together, and the kinetochore is the actual spot where it connects to the microtubule. So this is a question. A diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell in prophase 1 has, so in prophase 1, we know that there's it's diploid. So there's a set from mom and a set from dad. And there's 4 pairs of chromosomes. So if there's a set from mom and dad, there's 2 sets. You're obviously going to have 4, but there's also sister chromosomes. So there's going to be actually 4 sister chromatids. Why a fruit fly and cell in metaphase 2 has. So in metaphase 2, they've already gone through meiosis 1 and split apart. So we can see that once they've split, there'll actually be 2 sister chromatids left. Okay, now we'll go to number D. A blank test cross is a genetic cross that can determine if a purple flower pea plant has the PP or little PP heterozygous genotype. So in the test cross, there's it can be it's talking about the homozygous, or I'm sorry, the dominant trait or allele. So this is the dominant, and this is also dominant. The PMS, the little p here. And so in order to figure out which one of these is the parent trait, you're going to take a little pp, and that'll allow you to determine through the gametes which one is the correct parent. And that's the test cross. Okay, non-functional allele, sometimes called a null allele. This is an allele ultimately not the wild type, but a mutation that occurs, and this allele actually, its protein doesn't actually show the phenotype. So it's called a null allele, while a partitional and functional allele is called a hypomorphic allele. Hypomorphic is where it's kind of functional. Not all the protein is defunctional, but some has some functionality. So that's that. Now we'll go to number F. Mendel's law of segregation involves 2 traits. Mendel's law of independence involves a single trait. So the law of segregation is talking about the original homologous chromosomes in meiosis 1. And it's where this would be where one trait would meet and it would replicate. There would be 2 sister chromatids to the homologous chromosomes, and they would separate during anaphase 1, and it would be segregation, meaning that the 2 traits that are involved will ultimately determine the gametes. Now, Mendel's law of independent assortment, this is where in prophase Pachytina, I think it's called. There'll be independent assortment. And what this implies is that the homologous chromosomes with the sister chromosomes, they can mix and match. And so it's not going to involve two traits, but the single trait can actually mix and match depending on the traits or how they put it together, and that involves a single trait. Okay, and that explains the fill in the blanks. So now we'll go to number 3. I'm going to pull it up. Okay, here we go. All right, here we go. All right, I'm back. Okay, so number 3. Suppose an organ has a genotype TT, big R, big Y, little y. Assume that TT and little t alleles, the R, little r alleles, and the Y, little y alleles assort independently. So we're talking about independent assortment. Okay, so once this diagram, all the possible homologous chromosomes, arrangement in organism during metaphase 1. Be sure to indicate which alleles are found in the pair of homologous chromosomes. So we're talking about metaphase 1. So because of independent assortment, we can see that this is metaphase 1, and the purple is the homologous chromosomes. Also the reds and the Ys. And there's going to be big T, little r, big R, and little r, big Y, little y. So that's the genotype of the organism. And you can see the homologous pairs can sort independently. So we got big T and little t. And ultimately, big T's can go. So here's the homologous chromosomes, and they can go with big R, little r. And TT can also go with big Y, little y, independently. So that's the mix and match there during metaphase 1 after independent assortment in prophase 1. And then you can also have big R, little r, which can go with big Y, little y, and little r, big R can also go with little, big R can go with little y, big Y, and little r, big R can go with little y and big Y. So all these can be interchangeable, and that's what this is right here. Okay, number B, indicate all the pulsal organoids genotypes that can be produced in an organism. Conclusion of mitosis 2. So I drew this one out on paper. It's on a screenshot, so I'll bring you there now. So 1 moment. Okay, here we go. So we can see right here, these are the chromosomes. So we have mitosis 2. So there can be 4-8 Ts, 4-8 big Rs, 4-8 little Rs. I'm sorry, these aren't 8, these are actually chromosomes. So 4 chromosome Ts, 4 chromosome 12s, big Rs, 4 chromosome little Rs, 4 chromosome little y, 4 chromosome big Y. And back to the top. Okay. So that indicates organisms at the conclusion of meiosis 2. The gamete genotypes, which gametes are possible, and how many sister chromatids are found per cell in the organism during metaphase 1. There are 12. Because of replication, ultimately, you're going to have 2 big T, little t, plus big T, big T because of the sister chromatids, big R, little r, plus their sister chromatids, big Y, little y, plus their sister chromatids, and that gives you 12. How many chromosomes are found per cell in the organism during metaphase 2? There are 6. Because the homologous chromosomes have separated. It's asking for how many chromosomes per cell. And during metaphase, these are obviously 6, so they're going to line up, and it's going to be 6 chromosomes. Okay. So now down to question number 4. Okay, here we go. Number 4. A horse has 32 pairs of chromosomes. Did this one in my head, so how many chromosomes are present per horse cell at the conclusion of meiosis 1? So there's ultimately 32 pairs of chromosomes between the diploid set, mom and dad. And so with all those homologous chromosomes, obviously they're going to have sister chromosomes, which are going to be there. So when meiosis 1 occurs, they're going to split, which is going to be 32. So in each cell, there's actually going to, in each horse cell, there's going to be 32, which is 2 cells. So how many tetrads are present per horse cell during prophase 1 of meiosis? There'll be 32. So we have prophase, the meiotic spindle comes out. The nuclear membrane starts to break apart, and then you're going to have during the 2 prophase events, during those 2, I forgot their names. So they're going to come up, and there's going to be 32 tetrads because that's talking about when this synaptic complex comes together, the protein, all the homologous chromosomes will come together, and they're going to form 32 tetrads, referring to the sister chromatids. So there's going to be 32 tetrads. Ultimately 64 chromosomes, but 32 tetrads. So how many sister chromatodes are present in a horse cell during prophase 1 of meiosis 1? Prophase 1 of meiosis. So during prophase 1, there's going to be 32, because there's going to be ultimately 64, 32 homologous chromosomes in the replication, which is 32 present. How many sister chromatodes are present per horse cell during metaphase 2 of meiosis? So sister chromatids are present during metaphase 2 of meiosis. So they've separated the homologous chromosomes, and they're in each horse cell. When they go to the metaphase plate, the sister chromatids will actually be 32 present per horse cell, because they split during meiosis 1. And there's going to be 32 in one and 32 in the other. How many bivalents are present per horse cell during anaphase 2 of meiosis? So bivalents are referring to the homologous chromosomes. During anaphase 2, they're actually going to break apart. And the thing is during meiosis 1, they had split the homologous chromosomes. So there actually is not going to be any bivalents, because bivalents are referring to how many our homologous chromosomes sets are there. So there originally would be 32 in meiosis 1. All right, now on to number 5. All right, here we go there. All right, last problem. All right. All right, so this is the tetra hybrid cross and pea plants. Here's all the different alleles, the genes of the different alleles of the genes. They're mismatched. You got this in the original individual and the other individual. And so we have all the characteristics, and so we're doing independent assortment. So what you're going to do is you're just going to do individual Punnett squares for the T's, the R's, the Y's, and the A's. And so you can see down here it's rather small, but this is what's going on in order to fit it into the screen share. So what portion of the offspring be tall green? So we're looking for these characteristics. And we're looking for the phenotype, not the genotype. So you just do the individual Punnett squares, and you go to the dominant and the recessive, and you add all these together, and you're going to get 9/64, which are going to be these. You divide that as 4.1. Same song with this. Genotype's going to be, so we're looking for the genotype in this one. Genotype's not the phenotype, so we add up the genotypes, which is not the dominant mass or the homozygous recessive traits, and So we add them together. So it's going to be 1 over 32, and you're dividing, which is going to give you 0.3.1. This is heterozygous. So now we're adding up all the heterozygous for all 4 traits, all these, and we're going to get 1/16 or 0.2. A proportion will be dwarf wrinkled, so now we're looking for all recessive traits of the phenotype. We add them together, and we're going to get, here, let me pull it up. It's actually 0.2. You can't see it on here because it's down here, but that's the solution. And that ends this broadcast. All right.